Introduction. Hello, everybody. Yes, the topic of my presentation today is Marinosolv, our technology for enhancing hardly soluble compounds. I'd like to start with a short snapshot of Marinomed. The company was founded in 2006 as a biopharmaceutical company. And we have developed two technology platforms. One is a technology platform that is against viral infections, viral respiratory infections, such as COVID. And for this technology, we have medical device products, such as nasal sprays, throat sprays, and lozenges. On the market all over the world. In 2015, we established the Marinosolv technology, which is now successfully clinically proven in a phase 3 clinical trial. And in 2018, Marinomed has established an IPO. So we are on the stock exchange market in Austria. And we have now around about 50 employees. Marinosolv is one of the basis for several better therapies. We can enable novel stable formulations based on a saponin micellar platform. We have 2 clinical trials already proven the technology, and it's suitable for highly hydrophobic small molecules and also small peptides. The most advanced asset is Budesonide, budesonide-containing nasal spray. I'm coming to it a little bit later. And for this project, we are currently preparing for filing for an approval. The technology itself is supported with a very strong patent family since 2017, right about until 2036. Well, Marinosolv works generally in 2 steps. We're always starting with an aqueous basis containing of saponins such as astin from horse chestnut or glycyrrhizine from licorice root, and mixing it together with dexpanthenol. And in this aqueous buffered formulation, we have a self-forming micellar structure. And in a separate part, we are dissolving the hardly soluble API in a solvent which is very good mixable with water. So it's never an oil-water-based formulation. It's always very, very aqueous, very good mixable, such as propylene glycol, ethanol, something like that. And then the dissolved API in the solvent is going to be added to the aqueous part. And then we have a very, very stable, improvable micellar structure in which we can really strongly improve the API concentration in an aqueous formulation. Therefore, Marinosolv shows several advantages and key USPs. For example, due to the higher solubility in an aqueous formulation, we can strongly reduce the dose of the API compared to suspensions, for example, because due to the dissolved API, we can strongly increase the permeation of an API and therefore strongly increase the bioavailability. The excipients we are using are well established and very well tolerated, either in local tolerance studies and as well, of course, during the clinical trials. Misdosing is one of the key problems when it comes up to using suspensions, because the patients have to shake the medicinal product before they use. They very often forget that, and then they do not receive the dose they should receive at each time. In this case, we have a solubility. Therefore, you can avoid this shaking, and you can also you can manufacture the product preservative free, because you are able to manufacturing in with sterile filtration, aseptic filling. Very key advantages, especially when it comes up to high sensitive tissues. And of course, due to the higher bioavailability, we have a faster onset of action. Here is only a small group of key compounds that we have already tested with thank you with this technology. For example, a lot of corticosteroids such as budesonide or fluticasone propionate, as well as mometasone, but also some cancer therapy compounds like patopanib or paclitaxel. We have also used what we've heard today, cannabidiol. Was also one of the compounds we have already tried, in which we could increase the solubility in an aqueous formulation of up to 300 times. Then we have a huge project on macrolide immunosuppressants, especially on tacrolimus, which is used very often also in the ophthalmic diseases. In this case, I will show you a little bit later some more detailed data, but we were increased the solubility in an aqueous formulation up to 1 milligram per milliliter, compared to the water solubility of tacrolimus itself is around about 3 microgram per milliliter. Here are some opportunities, what can be done with Marinosolv, and what we have are actually also already done. Well, most of the projects we are doing internally are locally applied, locally acting, such as nasal sprays, eye drops. We are also working on inhalation products, but in this case, liquid inhalation. And currently we're starting also with several of our clients on working on oral formulations, in this case, both liquid and solid dosage forms. Our technology is most developed for liquid dosage forms, but we are starting recently also with changing some kinds of the formulation in order to get also the opportunity to receive some solid dosage forms. And of course, we have also gained some data together with one of our clients for tolerability of the Marinosolv matrix in IV injection, for example, but subcutaneous injection is also an option for the technology. Now I would like to start to show you two of our case studies, case projects, in which we used Marinosolv and how far we could come with that. And I would like to start with Budesolv. Budesolv is our first corticosteroid nasal spray solution. Most of the people do know budesonide containing nasal sprays with the name Rhinocort from AstraZeneca. But other competitors are also antihistamines. Antihistamines are working very fast, but not very long. Corticosteroids are currently only available as suspensions. And the immunotherapy is something that is very rushed and a very tough therapy, therefore should be avoided as long as you can. So the key need, the key medical need, and the idea for us was to develop a product that has an early onset of action, that does not have to be used 8 or 10 days before the allergy reaction starts. Here is an example of how we started with preclinical trials. Marinosolv was either dissolved using Marinosolv or formulated as suspension as for the market product, or we used Renocord Aqua, so our originator comparator, and we applied it onto ex vivo, onto a porcine nasal tissue, and determined the concentration of the permeated budesonide into the tissue over a time period of up to 60 minutes. The determination of budesonide in the tissue was determined via HPLC-MS, and additionally in a side experiment, we used here one of our model compounds, because it's fluorescently labeled and therefore we could visualize it a little bit better. We made a very similar experiment. We dissolved this hardly soluble estradiol, which is fluorescently labeled, with a Marinosolv formulation compared to, we formulated it as a suspension. And as you can see here, the estradiol goes much deeper into the nasal tissue. And here on this side, we see here budesonide enabled in Marinosolv formulation compared to the originator Renocord Aqua. And in this case, we had here starting concentration of the formulation of 300 microgram per milliliters compared to the originator of 1.28 milligram per milliliters. So it's 4 times lower. And we still have a higher onset of action. We started with a much higher concentration after 15 minutes of incubation, and we even increased it even higher compared to Renocord. Ready? In an in vivo study, we worked on a murine inflammatory lung model where we induced inflammation with LPS. I think this was something that we have seen today already. In this case, we had 2 different settings. In the first setting, we had a pre-treatment before LPS induction of 3 hours. And then after 2 hours after LPS induction, we took a ponchoalveolar lavage and determined the TNF-alpha reduction. So here we use the placebo as the control. And then we have Marinosolv with a strong TNF-alpha reduction and compared to Renocord Aqua, which is, I think, around about 50% lower. And in the other setting, we didn't make a pre-treatment, but a post-treatment. So we had first the LPS induction, and then we started with treatment 15 minutes after the induction, after the inflammation, and had also the readout 2 hours after LPS. And even though in this case we could observe a reduction of TNF-alpha compared to Rhinocort Aqua, where post-treatment doesn't really seem to have any efficacy. Based on the preclinical results, we induced a pivotal double chamber, no, a pivotal double-blinded challenge chamber study, placebo-controlled and comparator-controlled. Therefore, we had volunteers that received in 3 treatment periods first either placebo, Rhinocort, or Budesolv. Budesolv was here at a concentration of 200 microgram per milliliters, and they came into the challenge, received started a challenge of grass pollen, received the first dose on day 1, around about 90 minutes after the challenge started. And then they continued the challenge for 6 hours. Then the patients received the medication to home and took it for another 6 days. So day 2 to day 7, 2 times a day in each nozzle in the morning, once a day. And then on day 8, they come back into the challenge, and then they received the challenge again. So we had as primary endpoint a therapeutic non-inferiority compared to Rhinocort Aqua. And additional, as key secondary endpoint, we wanted to see the onset of action on day 1. And all volunteers received each treatment, placebo, Rhinocort, or placebo. Therefore, we had also a control of each patient for each treatment. Here are the results. I'd like to start with the right one. This is the total nasal symptom score on day 1. And we see that we started with a change of the symptom score after about 2 hours, where the symptom score for Budesolv decreased compared to placebo and Rhinocort Aqua, where the symptom score didn't change really a lot. And additionally, here on day 8, we have a strong change of the total nasal symptom score for both Rhinocort Aqua and Budesolv compared to placebo, which means that we have met both the primary endpoint of non-inferiority, which gives us the opportunity to apply for application compared to Rhinocort. And additionally, we had met the key secondary endpoint of onset of action, which gives us, of course, a much higher bonus of the SMPC in the SMPC. The second case study I would like to show you is Tacrosolv. It's a tacrolimus containing eye drop formulation. Tacrolimus as topical treatment can be used for example, allergic conjunctivitis, in which there is only existing in Europe and in the USA, nearly everywhere in the world except from Japan. Only antihistamines and mast cell stabilizers. In Japan, there is existing a tacrolimus containing suspension called Talimus, but that's more or less. Additionally, there is the opportunity to treat dry eye symptoms, in which we have a hand of few competitors, such as Restasis, Icarus, but all of them are cyclosporine, and compared to cyclosporine, tacrolimus is a much more potent API. We have performed here, like for placebo before, an experiment with the model compound estradiol on porcine cornea. Therefore, we had in vivo studies in porcine eyes. Oh, I'm sad. You do not really see it. Yeah, maybe you can see it. If someone is interested, I can send you the original slide, then you can see it much more better. In this case, you can see that on the cornea, or in the cornea, tacrolimus or the estradiol was really good permeated compared to the suspension, where you just can see the estradiol on the surface of the cornea. And additionally, which is even more important, we also could recognize that the estradiol was even permeated into the scleroretinal layer, which you don't see when you formulate it as a suspension. And additionally, we treated the pigs 4 times for 1 day with a concentration of 700 microgram per milliliter of tacrolimus compared to thalimus. Thalimus has a concentration of 1 milligram per milliliter. And in this case, then we determined the concentration of tacrolimus in the cornea via HPLC-MS and found not round about 3.8 microgram per gram of the cornea. And in tacrolimus, there was really low value. An additional in vivo treatment was performed on a 4-day experiment in pigs, in which the pigs were treated 2 or 4 times a day with also 700 microgram per milliliter of Tacrosolv formulation compared to Prograf as an standard oral formulation. And in this case, you can see that we have a dose-dependent concentration of tacrolimus in the cornea, which compared to, let's say, nothing when it comes to oral treatment, which is not really a surprise. Based on the results that we have gained during the preclinical development, we strongly reduced the concentration of tacrolimus for a clinical trial. In this case, it was a phase 2 dose finding study. Therefore, we had 2 doses. So it was the same concentration of 50 microgram per milliliter compared to, let's remember, 700 microgram during the preclinical trial. So we decided to strongly go down. The 50 microgram per milliliter was given 2 or as 1 drop per eye per day. It's a very similar study to the challenge study you've seen before, except from we have here only a placebo and the tacrolimus and no comparator, because we were going on to a dose finding. We wanted to see where is the effective dose. And in this case, we received, or the volunteers received the first dose half an hour before they entered the challenge chamber, and they had to sit not for 6 but for 4 hours. We have seen during screening with the volunteers that 6 hours might be too long for them for suffering. Therefore, we reduced that to 4 hours, and they received either placebo or tacrolimus with a washout period of 3 weeks. And they also received the drops to home. They gave it themselves for day 2 to 7, and then they came back on day 8 again and repeated the challenge. And then we had here as primary endpoint the therapeutic superiority to placebo with any kind of the 2 doses. And as secondary endpoint, we wanted to see that we have a difference between the 2 doses. We wanted to see, do we really have a significant difference between the 2 doses? Here is what we can show you for the results. The study is not completely finalized at the moment, so the report is currently ongoing. But what we have here are the first key results that we have already published. On day 1, we do not see any kind of change for total ocular symptom score, but on day 8, we definitely see it for the higher dose. So what we can say now is, yes, on day 8, we can see with this very, very low dosage, significant difference to placebo for the higher dose, and we definitely also see a difference on day 8 between the 2 doses, which is very nice, but gives us also a hint for a further or upcoming study that we should increase a little bit the dosage, especially when it comes, for example, to other indications such as cornea transplantation. Then we should definitely increase the dosage. Here is another result, what we see after 1 week of treatment, that we have a significant change in total ocular symptom score when it comes to during the challenge at the last two time points, evaluation time points. Then we have a significant change compared to placebo with the higher dose. So, and this results gave us the opportunity to offer this technology also to our clients. Means that we are offering the technology for using their our technology in their API, either if it's new chemical entity or if it's a better formulation of a traditional API. Doesn't really mind for us. We are open for everything as long as it works with Marinosolv. And one of the first questions that we receive generally is, how much can we increase the solubility with my API? And I have always to give the same answer, I don't know. We just have to double check it. We just have to see it. We have to go into a feasibility study. But what we can say is that we have an excellent tolerability for oral and intravenous studies. We have reported data, therefore, which we can also share under confident. We have very good PK studies with a special NCE small molecule from one of our clients, especially when it comes to the comparator that they have also tested. I'm not allowed to tell you which one it is, but what we can say is that we have performed both repeated dose toxicity pilot studies as well as pharmacokinetic studies in Vista rats. And both are fine, so we are continuing the project, which is very nice. Formulation development with Marinosolv serves a lot of opportunity, such as we are not limited to any specific kind of API groups or any specific kind of APIs, as long as they are hydrophobic and hardly soluble. If you have an API that is really good water soluble, then it really doesn't make any sense to go with Marinosolv, because this is one of the key advantages of it. It is suitable for APIs, either if it's an old traditional off-patent form, if it's NCE or so new chemical entity. And we really don't mind if you come to us at the stage of reformulation because you want to improve your product, which is already on the market. If it's off-patent and you would like to increase new IP, if it's during life cycle management, or and we are very open to efficient drugs with low systemic off-target activity. So here we have a wide range of opportunities. Marinosolv for You partnership, we have already and offer project for locally applied, locally acting drugs for systemic therapies, oral, intravenous, subcutaneous, whatever. And shown the tolerability in preclinical and clinical settings is very good. We had nearly had all excipients that might be possible in several local tolerance studies or clinical trials. We are working most of the time on liquids, but we are also open for freeze drying. We have already performed some experiments with freeze drying, which were very successful. And currently we're starting with spray drying or with solid dosage forms, so it's all open. All formulations we are going to perform are generally customer made. So we are always trying to consider what type of indication is planned with this API, what type of application form is planned with this product. We are always trying to consider from the beginning the physical chemical characteristics of the API, trying to consider which pH might be the best for the application form, for the tissue that's going to be used, but as well as for the API that needs it, and trying to find out which is the best formulation for each API. So it's never only just, I give it to you and you're trying it by yourself, because it's always trial and error and trying to find out which combination of Marinosolv might be the best. And of course, we are assisting them not only during the first feasibility study, but also for later pharmaceutical development. We're trying to discuss together with our clients what kind of preclinical and pharmaceutical development experiments needed to be done. Would the client like to perform them by themselves? Sometimes yes, sometimes no. Or should we coordinate them? Should we conduct them? And at which stage we should try to discuss with their CDMO, or if they have their own manufacturing, we are also open to do the tech transfer very early. So this is a very strong communication with our clients all the time. And here's just a short outlook on how does such a project is going to work. We always start with a short feasibility study, and this feasibility study is taking around about 3 months. And in these 3 months, we are just trying to find out which is the highest possible dissolved concentration of the API in a Marinosolv formulation. Doesn't really make any sense at this point to ask what is the best concentration for the dedicated API, just trying to find out what can be maximum dissolved. And yeah, so after the 3 months, our clients get a report in which all the formulations we have performed are documented. And for example, also in this kind of study, we do not concentrate on degradation of or stability, because the time period is much too low. We're just doing really just a solubility study. In the step 2, we are going then deep into formulation development, into pharmaceutical development, starting with thinking of what kind of packaging should be needed, what kind of stability studies should we conduct, complete formulation development, and as well, also the analytical procedure is very important in this case, then trying to validate an analytical method, including all the degradation products, the impurities, trying to consider everything from the beginning. And then the next steps, then we are going into developing a manufacturing process, going into tech transfer, but it's also very, very in close discussion with our clients, how they would like it. And the last step in this case is always for us a licensing model, starting at the clinical phase. So if the product is going into clinical phase, then we are starting with a licensing model. So thank you. Open for questions.
A very interesting discussion and presentation of this technology that obviously serves as a kind of platform, as you mentioned.
Yes.
I would have some question regarding the systemic approach.
Yes.
Have you done some tolerability study, maybe even a feasibility study with some selected APIs already for the sub-Q and intradermal route? Because that seems to me also from a self-administration perspective something that is worthwhile to look into, especially when I look at what you claim here as a kind of patent life cycle extension.
For intradermal, we have currently not performed anything except from some internal, but really only internal experiments without any data that we really can disclose. What was the second question? Sorry.
Well, actually, sub-Q, intradermal, whether you have explored that, whether you generated some data in terms of tolerability, for example, in the guinea pig, that you have this kind of fundamental data, and maybe even some drug-related feasibility study results.
We have performed some local tolerance studies, but only for nasal and eye. And one of our clients has performed the oral and intravenous, but also IP. So intraperitoneal. But this is, in this case, what our client has performed is no data that we can show. This is just the only that we can, but we can ask him if we have a specific question, then we can show it to him. And that's it. Sorry.
You mentioned in one of your slides that this technology allows no system of targeting activity.
Mm-hmm.
Can you elaborate, please, how improving solubility can avoid the off-target activity?
I think it's here, right?
Yes.
Yeah. The key advantage of Marinosolv is that we can strongly reduce the dosage of the API compared to what's currently on the market, for example. Therefore, we have very strong local activities for when it comes to locally applied, locally acting. But in this case, when we are going into, for example, locally applied, locally acting, then we can reduce so strong the dosage that we do not really have any systemic activity.
The systemic activity will come from absorbers, and usually the product is more absorbed because it's more soluble. Because usually the
permeation is higher,
exactly
yes.
higher. So I don't see how when we improve the solubility, improve the availability, we reduce the on-target. So that's somehow contradictory.
Yes, that's right. But the excipients that we are using do not really give their own activity.
Like an excipient. So the target of target is for the API, it's not for excipient.
Right. Yeah. Does that help you?
Still not clear, but
Okay.
I appreciate it.
You had one.
Yeah, just wondering what the stability of those two case studies.
Yes. Budesonide is an API that is not stable against oxidation, but we were able to stabilize the formulation as much as we can in order to have more than 36 months stability at 2 to 8 degrees in cooling, and around about, I think it was now 9 months at room temperature. And tacrolimus is very tricky. Tacrolimus is unstable when it comes to hydrolysis, which is a bit of a problem when you have an aqueous formulation. Therefore, we have a stability of 9 months at 2 to 8 degrees currently during for this study medication for the phase 2 trial. But we have other APIs, for example, for fluticasone propionate. We have currently a stability of, I think, 24 months or something like that at room temperature.
But after first use, the shelf life is like 30 days? Or?
No, after first use, the shelf life doesn't really change for budesonide. For the Budesonide project, if you're taking it then at room temperature, you can use it for at least 3 months at room temperature. For tacrolimus, we had the SMPC for the study medication as using it for a 2 to 8 degree, and after first opening, in this case, we used single use, but if the study medication was stored at room temperature for a week, then it was fine. So we had already data also for room temperature, but just a few days.
It's not final formulation.
Yeah,
So because
It's definitely not the
when
final
we-
formulation.
when we're moving the formulation and for the next clinical stage, the formulation will be different and more stable.
Because these both are sterile products, right? So the eye drop is sterile and the other one is also.
Eye drop has to be sterile,
Yeah, exactly.
always.
So I think that there's when it comes to after first use, usually for sterile products, you're only able to use it for 30 days because then you might lose.
For nasal sprays, you do not have to be sterile. You just have to use an aseptic filling if you are not using preservatives. And if you are using then special nasal spray devices that has a preventing filter, then you can also use it much longer. But you have to take care of when it comes to the primary packaging. If you're going with a nasal spray without these preventing filters, then yes, you're right, but then you have any way to use preservatives. When you're using this special pumps, then you can also use much longer in uses. Because you just have to confirm the data then. And when it comes to eye drops, it's even more complicated because eye drops always have to be sterile. And in this case, you either start with single-use eye drops, then you anyway can use it for once. Or you're using preservatives in this case. And additionally, there are a few primary packaging devices that have these preventing filters as well. And then you can use the formulation much longer.